I'll be talking today. I'm Lorenzo, and I'm the head of Vascular Cell Dynamics Lab at King's College London. And I want to talk to you today about how we use high content image analysis. And then we turn this data into multiscale simulation to investigate the emerging cellular heterogeneity in endothelia. So first of all, some quick facts about endothelial cells, which are omnipresent through all mammalian tissue, and they contribute to tissue development and homeostasis. And give me a second here. And sorry, endothelial cells represent, they are 20-25% of all nucleated cells in the human body. They are only outnumbered by erythrocytes, which are not nucleated. They are the inner lining of all blood vessels and the main cell type in capillaries. The cardiovascular system, which includes endothelial cell, is the first functional organ system in the human body during development. The endothelial cells are the main cellular player during vascular angiogenesis, which is the formation of new blood vessels. They are the key players during tissue development, and they are the gatekeepers of inflammation and immunity, and is being shown in many articles and reports, which that localized endothelial cell dysfunction is a hallmark of several pathologies, including cardiovascular disease and cancer. And you see here in this nice image where Bell and colleagues stained human embryos, and they for different markers, including some of endothelial cells, and show how many and how important is the vasculature and endothelial cells in the human body. And this is an example of what happens in the lung, where we also have some heterogeneity. In regards of endothelial cell heterogeneity, it has been shown that endothelial cells are phenotypically, transcriptionally, and functionally heterogeneous across different tissue and within the same tissue. This is the work from Kaluchka and colleagues, which is published on Cell in 2020, where they have done single cell RNA-seq of mouse tissue, mouse endothelial cells, and they have shown that indeed, endothelial cells in different organs are different transcriptionally, and then when they subset, they pick endothelial cell from specific organs, and I have chosen the brain and the heart as prototypes here. They are also heterogeneous, and this heterogeneity might be arteriovenous specification, they may be lymphatic specification, but we are also starting to find that within the same vascular bed, we have heterogeneity. So our aim in our lab is to understand the molecular foundation of this heterogeneity and how this is dynamically regulated at multiple scale during development, homeostasis, and the functional consequence of this differential regulation. Because this is the key to understand a little bit better human physiopathology across the lifetime, because we know that endothelial aging is one of the hallmark of aging. To develop more effective treatments of life-threatening disease, cancer, cardiovascular disease, as I discussed before, and to develop innovative tissue engineering strategy to try to address these problems. So in my lab, we use primary endothelial cell culture, and we analyze them by high content image analysis. Here are some examples, but I will show you some more things about that later on. Then we use three-dimensional functional assays. In this case, I'm showing you UVEC, treated or not with the APT, a Notch inhibitor, and we will discuss Notch a little bit more later on. And then we have started developing multiscale cellular automata models, which help us figuring out how perhaps this molecular crosstalk between neighboring cells is affecting the cell phenotype. And after we try to discover the most that we can out of this data. And then one other aim in my lab is to actually use this knowledge to develop better protocols to differentiate and to obtain endothelial cell, specialized endothelial cell from stem cell, either pluripotent induced stem cell or, well, mostly pluripotent induced stem cell. Let me see if I can get a pointer, because I'm not sure that you see my pointing around. And so the first qualitative observation that we made when we observed the culture endothelial cell from different territories, here I'm showing you UVEC, human umbilical vein endothelial cell, and human pulmonary microvascular endothelial cells, or UVEC treated with DAGF, the prototypical angiogenic molecule. They are heterogeneous in shape, and for the sake of this talk, in NICD, which is the second messenger of notch signaling, and also downstream notch effectors. So UVEC at baseline, untreated, can display different level of NICD, and it increases in DHF-treated UVEC. Surprisingly, we find a microvascular pulmonary microvascular endothelial cell display high level of notch signaling, and aortic endothelial cell frequently display medium high level of NICD, and this is expected out of the literature and current understanding of the system. And in all endothelial cell monolayer we observed, many cells display very low NICD level. And this is not really expected, or at least is not being reported previously. So starting from this qualitative observation, we start trying to figure out what happens, why this is happening. On the right here, you see some images that is not mine, is not from our lab, but is out of this paper from Coet and colleagues, where they perform M-phase staining of human aorta or other big blood vessel in mouse, and just qualitatively can appreciate that, at least in shape, it's obvious that the same heterogeneity is also relevant in vivo, which is obviously very important. So coming back to the work of Kaluchka and colleagues, we explore their own database, which is available online. And if you want to know more, you can go at this website. And we were interested in notch signaling, and we immediately wondered if, okay, we notice notch heterogeneity in vitro, will this be the truth also in vivo? And actually, if we look at these plots, which are Disney plots of the single cell transcriptome, in the particular, we picked the pulmonary endothelial cells. It's valid for others, but here for just the illustrative purpose, I'm showing those. And if you look at the dots, they are obviously heterogeneous. So HES1 and HEY1 are downstream effector of notch, and the cells within the same lung are very heterogeneous in these genes. So this gives us some strength in thinking that the heterogeneity we find in vitro is also very relevant in vivo. So we have some question here. So what is that cause the intrapopulation and cross-population heterogeneity in notch signaling? And we also wanted to try to understand whether this level of heterogeneity is biologically relevant and how. Our hypotheses are that the intrapopulation heterogeneity in shape can be due to proliferation, in notch can be due to lateral inhibition, lateral induction, or all of the above. We really don't know, but we have tried to develop tools to understand that. And for across population, we know, at least has been reported, that differential baseline level of delta 4, notch 1, 4, and jagged 1 can cause this heterogeneity or this differential regulation. About the second question, if the level of heterogeneity is biologically relevant, well, the answer we think is yes, because it can mediate and dynamically regulate specialized function within the same vascular territory. And for example, functions that are important to endothelial cell homeostasis are both proliferation and junctional stability. But as cell biologists, we all know, these functions tend to be quite opposite. And so since they happen all together, they need to happen all together, we would like to understand why and how. So how can we investigate this hypothesis? We developed an high content image analysis tool, which allows us to quantify spatial heterogeneities within cell in the same monolayer. We developed this tool by using many of the tools we have discussed before, we heard before in previous talk, Fiji, ImageJ, which help us very much for machine learning aided pixel level image manipulations, and also for some cross-platform image input-output. Cell profiler and cell profiler analyst, which are very good at object segmentation and single cell data extraction, while cell profiler analyst is very powerful for machine learning guided object classification once we get information about these cells, this object that we measure. Then we use RStudio for statistical analysis. It's very good for data input output, large data manipulation, statistic plotting, interactive visualization. And finally, Computational 3D, which is an SBML compliant multiscale cellular automated simulation environment. So here is a very quick benchmark, a little bit arbitrary, and we benchmark our ECPT against some other software, which is commercially available. And the one thing we found, at least we think that our system is not necessarily much simpler than out-of-the-box software. Actually, you need some proficiency in these tools to be able to use ECPT, but this little effort is compensated by much more power in manipulating images, and we all know that Fiji images are very powerful at that. For which regards object and object classification, the simplicity of the ease of using our ECPT is more or less the same that we have in commercial software, and again the power seems to be way stronger. In terms of data analysis, the simplicity of R, once you are into the loop, or once you understand how to use it, is simpler perhaps, and way more powerful than any out-of-the-box system. And finally, in terms of simulation, our ECPT directly interface with Computational 3D. And this is something I personally haven't seen before in any other commercial or non-commercial package for high content image analysis. So what we use this for, to answer our question, or to try to answer them. And so question one, what causes intrapopulation and cross-population heterogeneity in cell area? So we've seen that happens both in vitro and in vivo. What could it be? One hypothesis is that this is linked to cell cycle, and is a kind of easy hypothesis to make. And our endothelial cell profiling tool, among other things, allow us to capture the cell state of the cell cycle state of the cell we are measuring. And these plot are representation of cells which are in G0, G1, or in S phase, or in G2M phase, and late mitotic. To clarify what late mitotic, what we mean for late mitotic cell, we are talking about these cells here, which are obviously they just divided. We notice it from the chromatin condensation and the fact that they are detaching one from the another. And just trying to link from some previous talk, what we find is that these cells tend to be kind of isolated. And so we are puzzled by this, because there is a whole idea that endothelial cells, they self-renew by some kind of stem cell present somewhere in the body. But this matter is controversial. And so how do endothelia self-renew? This is very important because it's connected with aging, and with vascular disease, we know that is very important for many pathologies or aging. So out of our data, we are starting to challenge the hypothesis that endothelial stem progenitor cell, they actually exist, but if they do exist, are they actually the main player in self-renewal of endothelial cells, or they are backups, or they're just bystander, or else? So the one thing that it seems evident to us is that endothelial cell, almost all endothelial cell within a monolayer cultural endothelial cell can proliferate. So without the need of a particular specialized stem cells. So then we move from the heterogeneity, intrapopulation heterogeneity in notch signaling. Well, both intrapopulation and cross-population heterogeneity. Regarding the cross-population heterogeneity, we measure the notch signaling in aortic, pulmonary, and UVEC. And we see this is just the empirical cumulative distribution function representing our notch level in our system, in our experiment. And we see that these are different. The blue are the UVEC, the red are the aortic, and the green are the pulmonary infravascular endothelial cells. But this plot do not allow us to actually capture spatial correlations between the cells. And since we are looking at cells in phase, we really want to understand how they spatially correlate. In particular, whether notch signaling might be similar to this pattern in the center, or might be more random or clustered, as also been shown in one previous talk. For this, we use one index, which we have implemented in our endothelial cell profiling tool, which is the Moran's index for spatial autocorrelation. In very brief, Moran's index of very positive Moran index will indicate cell clustering. Very negative, close to one Moran index will indicate cell sparseness, so a checkboard appearance of the different phenotypes, or random. 0 will indicate a random distribution. We then apply this to our maps of endothelial cell, which we extract via ECPT, and we find, again, surprisingly, to some extent, that there is no salt and pepper checkboard patterning, which is what the theory would suggest, because we know that notch is typically working by lateral inhibition, but also there are other hypotheses whereby we can have lateral induction. So to try to understand and to demonstrate this hypothesis, so whether it's lateral inhibition, lateral induction, or other mechanisms, we have started developing computational methods, because the problem is that it's very difficult to image and to segment these cells live. And so we need some theoretical framework to make some hypotheses and to try demonstrating these hypotheses. To do that, we integrated the Computational 3D, which is being developed by Prof. James Glazer at Indiana University. And we essentially extract our ECPT data and cell maps and their correlated data. We move them to Computational 3D. We create, simulate. We have created simulation of notch signaling in endothelial cells. We create synthetic data, and then we go back to R and try to figure out what kind of simulation can represent our data. And these are some very important topics that needs to be taken in consideration when developing computational model, cellular automata model. So the models need to acknowledge and account stochastic phenomena, or at least compensate for those, because we have very little information about that. They need to account the dynamic process, and we have to understand the time scales of this dynamic process. They need to be multivariate and based on as much as possible on real data beside the theory. So this on the right panel is the kind of phenomenon we wanted to implement in our simulation, which is cis and trans notch signaling due to different ligands, also including some ligand discrimination. So we developed the mathematical frameworks with ordinary differential equation models. This is just a stub, a very implicit way of describing this. Then we validated our model, trying to figure out whether this was overlapping with previously described model, for example, that described by Boareto and colleagues on PNAS in 2017. And we simulate our one cell dynamics. And well, believe me on the word, it seems that really we are reproducing the data that have been proposed before. And then the next step, which is a little bit more complicated and where we really need the CPT or similar tools, where we want to spatialize the model and we want to actually look at multi-cell dynamics. And this is what we are doing. And here, when you see our cell moving, you are seeing some little animations regarding our system. And if you see in some standard condition, this simulation is showing us on the right, is showing us that indeed, at least in certain conditions, we achieve and we obtain the typical salt and pepper pattern. So what is that models and the simulation tell us? So the formalization, the mathematical formalization allows appreciating some alternative hypotheses. So this that you see on the right is the standard hypothesis in the field. So the salt and pepper lateral inhibition patterning. Simulation also justify possibly expensive testing of alternative hypotheses. So if we don't find the standard pattern, and we obviously need to go in live imaging and demonstrate directly that this is the case. And so, but this might be very expensive. So having some support, theoretical support, might be very useful. Simulation might save us from pitfalls in drug discovery and unexpected side effect, because once we have a working simulation, we can really try to drug it, so modulate, make perturbation within the system, and actually try to see what happens to our phenomenon of interest. And so this simulation can really speed up drug discovery and recover the initial investment, perhaps in testing the alternative hypothesis. And while developing this simulation, sorry, we start understanding a little bit more the foundation and mechanism of cell biology. At least this is my personal experience. So here, going to some data, we have implemented our simulation, we have created it, we have run it, we have run a so-called parameter scan. In our case, we just scanned 4 parameters, which is more than enough, because out of these, we generate at least 865 combinations, and we didn't do a very deep parameter scan. And the very interesting and perhaps not surprising finding is that knot system, without huge modification to the theory, no, actually without any modification to the current theory, can generate qualitatively different response, some of which weren't really expected. So some responses are mono or bistable. So for example, if you look at the green patterns here, we see that we have 2 peaks in our density distribution of high and low notch cells. And this is corresponding to the salt and pepper distribution. And we can find some monostable system whereby we have very narrow banded knot signaling. So where we have essentially all cell having more or less the same level of knot signaling. So at when we look at the image, they are essentially all the same. But then we have other patterns, which are the more nuanced one that we see in the yellow and red panels here. And these are very interesting. They are unstable over time. So one of the first findings is that essentially, when we find certain patterns, these cannot be stable. So one cell which pertains to this kind of patterns, the cell won't stay in the same signaling status over time, and it will change dynamically. So this is very interesting because for reasons I will discuss in the next slides. So another important thing, and this is
Lorenzo, sorry to interrupt. We're way over time.
Oh, so okay,
if you can, please try to wrap it up. Yeah, I
will wrap it up immediately. So irregular cell disposition also dampen the system. We have fitted our experimental data, and we try to estimate time scales. We have found data that actually fit the experimental data, and we found patterns that look exactly to the cells that we look when we look with TCPT, and we confirmed that these should be oscillatory. So the important message here is that dynamic regulation of phenotype increased adaptability and plasticity of cellular response. And so endothelial cell in the monolayer can acquire differential phenotype to decide whether to proliferate and to expand the repertoire of possible cellular response. And so here to really wrap up, I won't repeat many of the things, but I would like just to point you to some of the literature which open the ways to this kind of investigations, like the work from McCarron and Mack, and our own work on the ACP, on the development of ACPT, which you can find at this, which has this title, you can find on Berkside, and this been published there in 2021. So I would like to thank you very much. Sorry for being over time. And this, I would like to thank all the people that have contributed to this work, the ACPT team, the other members of Vascular Cell Dynamics Lab in 2021, our collaborators, the Stem Cell Hotel at King's, Prof. Jane Glazier and the Computational team, and Matteo Battilocchi at the CSCRM at King's. And thank you very much for your attention, and I'm happy to answer any question if you have some. Thank you.